The overall goal of this procedure is to rapidly screen a large number of host plant volatiles for potential biological activity. This is accomplished by first collecting and analyzing by GCMS the host plant volatiles of interest. Then, the chemical components are identified and verified. The third step is to screen the individual components in sufficient replicates for bioactivity via electroantennographic analysis. The final step is to formulate and evaluate blends of active components. Ultimately, results can provide a truncated series of components or blend of components ready for further investigation in behavioral studies with the insect of interest.
The main advantage of this technique over existing methods, such as time-intensive behavioral studies, is that rapid screening by electroantennographic analysis significantly decreases the number of compounds needed to be taken on for other studies. Demonstrating the procedure is Mr. Wei Ji, a technician from my laboratory.
Once the relevant volatiles have been identified by gas chromatography and mass spectrometry, it is possible to perform electroantennography analysis, or EAG, of each available volatile. First, however, the available volatiles must be prepared. Prepare a solution of each volatile in pentane at a concentration of 5 mg per milliliter in a container that can be tightly sealed and stored at 4 degrees Celsius. Next, prepare the stimulus pipettes. Label 10 Pasteur pipettes